Hi, Dr. Brian Kaufman, a retired family doctor, CLL patient myself, Executive Vice President, Chief Medical Officer at the CLL Society, here at ASH 2024 in San Diego, California.
Hi, Meghan Thompson from Memorial Sloan Kettering Cancer Center in New York, New York.
Dr. Thompson, one of the big unmet needs in CLL is for patients who relapsed or become refractory to prior therapies. And there's a new class of drugs that you're going to be presenting some important data at ASH that don't block BTK but degrade BTK. So first, before we get started, explain to me what this drug is and what the difference between a degrader is and an inhibitor is, because we know about ibrutinib, zanubrutinib, acalabrutinib, pirtobrutinib, but this is an entirely different way of blocking that pathway.
Yes, so we know that drugs like first ibrutinib, then acalabrutinib, zanubrutinib, pirtobrutinib, they all inhibit BTK. And what is BTK? BTK is within CLL cells a very key target that helps mediate what we say B-cell receptor signaling. Basically, B-cell receptor signaling is really important for CLL cells to grow and survive. So if we can turn off this switch in the signaling, it's been shown that this translates into clinical responses and helping effectively treat the CLL. With an inhibitor, you're shutting down that sequencing pathway. But what we've seen is that over time, there can be changes actually in the DNA of where the drug binds that prevents these inhibitor drugs from working. So there's a new class of molecules that actually degrades the target or gets rid of it completely.
Chews it up.
Yeah, instead of and simply inhibiting it. And this could be really critical and has been shown initially in the lab and now with this drug in patients and several drugs in this class that actually degrading or getting rid of the target, might be an effective strategy for these patients.
So tell us about the research that you'll be presenting, and this is an oral presentation at ASH, so it's a big deal because it's very limited oral presentations. So people think this is very important, and I'm one who thinks this is very important. So tell us about this data.
Yeah, so I'm going to be presenting data from the phase 1 Cadence study, and this study is looking at a drug called BGB-16673. And this is in this BTK Degrader class. So it's a drug that binds to BTK, but also binds something called E3 ligase. What does it mean? Basically, it brings the target and tags the BTK protein for degradation or destruction. So it's actually destroying the BTK target. And so it's an oral pill that patients take, and it was designed for the situation that you described, patients where other BTK inhibiting therapies are no longer working.
And we know that it makes a lot of sense for patients because patients, as good as the therapies are, patients have limited options, and we know how important this B-cell receptor pathway is. So if you can stay targeting BTK longer and run that longer, that's very exciting. So how did this do? These were patients who failed BTKs, or tell us what kind of patients you were looking at and what the results were.
Yeah, so in this phase 1 portion of the study, basically all of these patients had to be eligible, had to have 2 or more prior lines of therapy, and all had to receive a covalent BTK inhibitor if it was approved in their home country for CLL. And so the vast majority of these patients had received a prior BTK inhibitor. Many of them also received a prior BCL2 inhibitor, for example, such as venetoclax, and many also received a non-covalent inhibitor. Not everyone, but we have some patients who've had a covalent inhibitor, venetoclax, and a non-covalent inhibitor. So a heavily pretreated patient population, and most patients had actually discontinued a BTK inhibitor in the past for progression.
So a difficult group to treat.
A difficult group to treat. We also looked at the characteristics of these patients. Some had BTK mutations or those changes that prevent drug binding of the inhibitors prior to coming on the study. Some did not. Some had p53 mutations or deletion 17p or complex karyotype. Some of these high-risk features for CLL was the patient population.
So I'm eager to hear what the findings were. Tough group to treat, options limited. They burned through at least 2 drugs and sometimes 3 or 4 or more. So what did you find? I know the numbers are small, but tell us.
So overall, so this was a dose escalation study because it started as a first-in-human study. We found a high overall response rate. One of the things we saw was responses even starting at the lowest dose level. And so there's patients with ongoing responses even at the lower dose level. At the 200 milligram dose level, which has been selected for further study, and there's a part 2 of this study that's ongoing given the promising results, the overall response rate was quite high. We also have seen patients who've had a deepening of response rates over time. So the longer and select patients they were on the study, they've had a deepening of responses. We've actually, one of the unique things that I'll highlight, some patients have had, while the majority are partial responses or partial responses with lymphocytosis, the white count's still elevated, but the nodes are responding.
We see that with all the drugs that target BTK.
And we see that with the BTK degraders. While that's the majority of the responses, there are actually a small number of patients with 2 patients with a complete response or complete response with an incomplete count recovery. And so as we move with longer follow-up, it will be interesting to see how those responses evolve. But at this time point of the data cutoff, we've actually seen complete response.
Wow. So I have to ask you about safety and tolerability because that's equally important to efficacy.
Yeah, absolutely. So overall, there was what we call dose-limiting toxicity, or in the initial phase of the study, a toxicity where the drug needed to be paused, and that was a grade 3 maculopapular rash. So a patient had a rash.
A raised red rash,
and the patient stopped therapy and was able to resume drug. But overall, very well tolerated. The most common, we grade on clinical trials, we grade the adverse events and their seriousness, and so grade 3 is generally a higher level of intervention is required when this occurs. The most common grade 3 or higher side effect was neutropenia.
Remind us what neutropenia is.
And that's when your infection-fighting white blood cells called neutrophils are lower. So that's one thing we observed. Although we saw that only 1 patient had what we call neutropenic fever, or a fever when the white blood cell count was very low. There are other side effects that we monitor for, things like bleeding with BTK targeting therapies. There were 2 what we call major bleeding events on the study. And so overall, promising, although early days, very promising therapy and generally well tolerated among patients.
What about the cardiac side effects that we see with some of the BTKs?
Yeah, that's a great question. So BTK inhibitors, traditionally we have seen cardiac arrhythmias associated, in
irregular
particular
heartbeats,
irregular heartbeats, and particular atrial fibrillation is one, so an irregular heartbeat rhythm. There was actually no atrial fibrillation observed at this time point with BGB 16673.
So very promising, very early. Any takeaways for patients about this drug in particular and this class of drugs?
Yeah, I think that this is a very promising drug class. I'll emphasize that it is a pill-based therapy, and so in this study, various dose levels were explored, but so different numbers of pills, but a once daily dosing strategy of the pill. So very similar to other BTK inhibitor targeting therapies, but using another mechanism of action. And the hope is that some of these BTK degraders, including BGB-16673, may overcome some of the shortcomings, including the development of resistance seen with BTK inhibitors. So I would say stay tuned. I think this is a very important class of drugs. I also want to thank the patients who have participated in the clinical trial. Their participation really has helped to generate a lot of knowledge, and many of them are benefiting from ongoing treatment of the drug as well.
Well, thank you for the research you're doing, and I'm a big believer in clinical trials, so thanks to all the patients who jumped in. Thank you.
Thank you.